So my name is Esko, Oksanen, and I work for the European Spallation Source in Lund. And I'm a protein crystallographer, and I am responsible for building one of the neutron instruments for the European Spallation Source. And what we're interested in studying with this instrument is the structure of biological macromolecules at the atomic level. Now, the usual tool to study these kind of structures is X-ray crystallography. However, with X-ray crystallography, we generally do not see hydrogen atoms. And hydrogen atoms comprise about 50% of all the atoms in biological structures, and more importantly, they are often the ones that are involved in the function of these macromolecules. So the power of neutrons is in being able to unambiguously determine the position of these hydrogen atoms from crystals of biological molecules such as proteins. So we are particularly interested in understanding things like enzyme catalysis, how the biological catalysts can lower the activation barrier of reactions. We're also interested in ligand binding to proteins, particularly pharmaceuticals, how they bind their targets, and how this of course helps us to develop better pharmaceuticals in the future. And the third thing that is very interesting for us is transport of protons, of hydrogen ions, across biological membranes, which is the basis for all energy production in our cells. Now, this involves membrane proteins, which are particularly challenging to work with, and with the power of the ESS, we hope that we can expand the scope of neutron crystallography from what would be called simple systems to much more challenging and complex systems that membrane proteins typically are. And we hope that this will revolutionize our understanding of the mechanisms of biological macromolecules.